Hello and welcome to day 2 at the 2019 BioInvestor Forum. I'm Christine Corrado with Proactive Investors. Joining me right now is Aldo Carrascoso. He's the CEO of InterVenn Biosciences. Aldo, so great to meet you.
Thank you for having me.
Aldo, you have a very personal story we'll get into in a second, but why don't you first tell me a bit about the company?
So InterVenn is propelling glycoproteomics from mere diagnostics into real precision medicine tools. So there have been a tremendous amount of approvals over the past 20 years of these things called glycoproteins. So your standard diagnostics, for example, for ovarian cancer, CA125 is a glycoprotein. Your PSA for prostate cancer is a glycoprotein. CA, CA99, and the list goes on. HER2 is even a glycoprotein. So they've traditionally been binary diagnostic tools. Actually, they're also some of not the best tools because they have a lot of false positives. Looking at it from the glycoproteomic point of view, instead of looking at a binary type of situation, you're looking at gradations and directionality. So we're able not just to tell you the existence of, let's say a tumor, also we're able to stage you. So one of the interesting things we're able to do is understand even the origination of cancer, the stage of cancer, sometimes even some very rare ones. And of course, this is all powered by a machine called the mass spectrometer. And just by the very nature of me saying that word, everyone already knows, oh, you guys generate a lot of data. So we do use AI, not artificial intelligence, augmented intelligence. So we've got really good scientists that know what they're looking for. It's just they're combing through millions of data points.
Why is it important for companies to be using augmented intelligence?
Well, I don't believe that machines will replace scientists anytime in the near future. I think scientists know what they're looking for. They do. It's like looking at a needle in a haystack, except for glycoproteins, it's a needle in 15 million haystacks. If you find a needle, you can go through a haystack, right? So we just automate that process for them. So my background actually is not in life sciences. Background is software engineering, statistics. So when I met my co-founders, Dr. Carlito Librilli of UC Davis and Dr. Caroline Bertozzi of Stanford, their biggest problems wasn't really the science, because they were masters at it. They've been doing it for decades. They said, I need software and I need the way to analyze this thing called the glycoproteome. And I was like, what do you mean? So when you were watching them, they were analyzing cohorts of hundreds of people. They were one of the first people to really analyze large cohort of glycoproteomic studies, like for ovarian cancer, for drug discovery. Some of their workflows used to take months and months and months. Like they would generate the information and a postdoc would have to go and annotate each one. Over hundreds and thousands of those things. And when I saw it, I said to him, Carlito, so from 9 months, what if we can get your throughput down to 9 weeks? Because again, machine intelligence, computer vision makes sense.
Right.
He said that would be like, you would accelerate my thing for like 10x. So when our neural network went live earlier this year, from 9 months, it went down to 10 to the negative 3. So it allowed us to look at not just one indication, it made us look at multiple indications. So pancreatic cancer, liver cancer, immune response, so immune oncology, and the list goes on.
Well, let's get to that personal story I mentioned at the top. I know that your family's been affected by breast cancer. So, did you really kind of see that and really feel the need to actually create what you're creating right now?
So people often refer to me as a serial entrepreneur. I'm actually not. I'm a serial problem target. So problems keep finding me. I keep on getting pissed off and starting companies. This is my 4th company. I've had success in the past, but this was also the one that's most personal. So I often tell people that my family was decimated by cancer. It still is. It started with my mother, who succumbed to breast cancer in the 90s. When there was nothing. And then I continuously watch my closest relatives developed the disease. And what was so striking about it is we don't have the genetic markers. None of us do.
None of the BRCA genes or anything
I
like
don't
that.
have it.
Yeah.
My other family members don't. So the incredibly frustrating truth of not, led me to, of course, the steps of the labs of my co-founders, where they were explaining to me that, instead of looking at genomics, which is a probabilistic methodology, look at this thing called glycoproteomics, which is the disease itself. So it's no longer a question, will I develop the disease? It's a question of how bad is it? Is it very, very, very early? I'm not talking about early detection. I'm talking about very, very, very early. It means, can you intervene at that point? So when I read their first 10 papers, it would be the first scientific papers I would have read outside of college. So I was like, this looks like science fiction to me. And my main question was, why isn't anyone doing this?
Right.
Like someone who was I'm a professional skeptic. And essentially they said, it's actually not that easy. You've got these giant machines that spits out so much information. No one's built the software. No one has the data. No one can train anything on it. And I said, guys like me, yeah, you're one of the first guys. So that was 3 years ago.
Well, how far away are you from commercializing it?
So we are in clinical trials.
Okay.
Yeah. And our preliminary results are not just encouraging, they increase the performance of our test for ovarian cancer. So we're doing a multi-country, multi-site, multi-cohort analysis for ovarian cancer. So it's women presenting with pelvic masses, so ambiguous pelvic masses. So over the lifetime of a woman, 1 in 5 of them will present with an ambiguous pelvic mass. And out of the 1 in 5, actually 12 to 18% actually are malignant. And not all of them are ovarian specific. Some of them come from metastatic cancers like colorectal and lung cancer, which we are able to detect. So what we do is we are able to properly differentiate malignant and benign masses. So what are we selling? Peace of mind. Peace of mind. So our multi-country, multi-site study kicked off late last year. And yeah, our preliminary results look terrific. So,
Great to hear.
Yeah.
Well, looking forward to following the company further. Really great to learn more about your background and the company. Aldo, thank you so much.
Well, thank you for having me.